Hello, everyone. Thank you for joining us today. We're going to give people time to join us while we are waiting. If you want to let us know where you're joining us from using the chat feature, that would be great. Hi, Liz. Okay. I'm going to give one more minute for people to join us. Hi, Karen. Hi, Joanna. Okay, I guess we could get started now. Thanks everyone for joining us today and welcome to the first lecture of the 2024 Childhood Cancer Speaker Series. I am Vidya Ganapati, Research Program Director at ALSF. Quick programming notes. Your feedback is very valuable to us and we would like to hear from you on our 2024 lecture series. You will have a follow-up survey launch when you leave the webinar. Please take a few minutes to fill this out. We have a total of 4 lectures scheduled for 2024, including Dr. Fox's lecture today. The details of the upcoming lectures and the link will be posted in chat soon. Finally, you can ask questions following the lecture using the Q&A feature at the bottom of your webinar screen. Without further delay, I would like to introduce today's speaker, Dr. Elizabeth Fox, who leads the regulatory and clinical trial operations as well as clinical research prioritization and strategy at St. Jude Children's Research Hospital. Nationally, she is the Vice Chair of Developmental Therapeutics for the Children's Oncology Group and the UM1-funded Pediatric Early Phase Clinical Trials Network. Dr. Fox received her medical degree from Pennsylvania State University and Masters of Clinical Research from Duke University. Her fellowship training and early career in pediatric oncology and drug development was at the pediatric branch of the intramural program of the NCI. Prior to coming to St. Jude's in 2020, she was a professor of pediatrics at the Pullman School of Medicine at UPenn, director of developmental therapeutics and oncology at CHOP, and she was also the PI of ALSF Center of Excellence in Clinical Research and Pharmacology at CHOP. Dr. Fox has contributed to the FDA approval of 7 of the 50 oncology drugs currently approved for children with cancer. Her talk today is titled The Pivotal Role of Clinician-Scientists and Academic Clinical Trials in Drug Development for Childhood Cancer. Welcome and thank you, Beth, for sharing with us today. The podium is all yours.
Thanks so much. Can you hear me okay?
Yes.
Perfect. Well, thanks so much, Liz and everyone at Alex's for the invitation and ability to present this, and everyone for your time in joining us today or looking at this recorded video in the future. This is something that I've been thinking about and working on for a long time, and I really am at a point where it is about the partnerships that we have. And I'd like to highlight today how those partnerships work and use an example of how a partnership that started with Alex's Lemonade Stand Centers of Excellence really demonstrates how to do this well and call all in our clinician scientist role to really advance the what we need to help children with cancer. And so I'm going to ask a question about about a third way in, and we may be able to get your responses in the chat. What I'm going to be asking about, so you can start thinking about, is I'd like to hear a little bit about your impressions of different sponsors for trials, whether they be industry sponsored or investigator initiated or cooperative groups, and how for you they have differed. Hopefully by the time we get to that slide, you'll understand. If you aren't familiar with those types of sponsorship for trials, you'll be all caught up and ready to go. So with that, I'll get started. And I have no financial conflicts of interest to disclose, but I do need to say that the views that I'll present do not represent the official views of institutions, cooperative groups, or other organizations with whom I may be affiliated. So what I hope to do in the next hour or so is first and foremost leave some time for questions at the end, but just frame the challenges of clinical drug development for children with cancer. Many on the line, I'm sure, can share their experiences with what are the challenges and what are the opportunities. I'd like to discuss some examples of successful and learnings in drug development across the years and where we're at currently and how to think about this as a way forward. And finally, the considerations that we as academic clinicians and others in the field who are committed to drug development for children with cancer, how can we meet the unmet needs for childhood cancer? And so one of the real advantages of thinking about childhood cancer is no matter who you are, no matter what part of the field you're in, you understand the unifying goal of childhood cancer drug development. It is to improve cure rates. And this is a bar graph that of 5-year survival across the decades. And what we know now is the 5-year survival for children with cancer is over 35%, over 85% for children who are newly diagnosed with cancer in the United States today. That is a phenomenal thing. We also know that 5-year survival is not necessarily cure. What we know is that those cures have come at a considerable cost, both in acute and late side effects. And so we'd like to diminish the acute toxicity. The pie chart in the middle is taken from a recent rhabdomyosarcoma upfront study where we accepted the fact that nearly 3/4 of the patients on that study were going to have a grade 4 or life-threatening toxicity during their therapy. And we think that that is if we can avoid that, that would be fantastic to avoid the insults to organ function and other parts of growth for our children with cancer. And finally, as have been well published by many, including those some on the line, we know that the late effects of our childhood curative therapy for childhood cancer are significant. This is just one example from Greg Armstrong through the childhood cancer survivorship studies that demonstrates very clearly that survivors of childhood cancer, 40% of those survivors have a grade 3 to 5 chronic health condition, so a serious or life-threatening chronic health condition if they are survivors. And that is compared to their siblings who have under a 20% risk of those same side effects. So this is a unifying goal. Also important to remember is that childhood cancer is a rare disease. All of childhood cancer on whole is a rare disease. There are only just under 16,000 children who are newly diagnosed with cancer every year in the United States. I agree that that's too many children, but compared to adults who are newly diagnosed with cancer at a rate of 150 adults for every child, this is an important consideration when we think about the partnerships for drug development, particularly the partnerships we have with pharmaceutical companies. What's also important to remember about childhood cancer is here in the table. This is a list that corresponds by color to the pie chart. And you can see that, as we know, acute lymphoblastic leukemia accounts for 20% of the children with newly diagnosed cancer each year in the United States. And as you can see, the list goes down with different types of cancer. Importantly, here at the bottom, there's a category called Other that accounts for almost as many as children with acute lymphoblastic leukemia. And this is a wide variety of different types of cancer. What's really important about this table is the fact that not only do we have different histologies, cancers, and ways to treat it, but the age groups that these cancers affect is predictable based on what we know about the cancers, and that also impacts drug development. So I want to start with where we're at. And this is a a graph from the adult National Cancer Institute match study, which is molecular targeting. And this was the first broad scale to understand what the different mutations are and can we match them to sub-protocols. In this adult study, they enrolled almost 6,000 patients and who submitted tumor to determine what the actionable mutations were in those patients' tumors. And 38% of the patients who submitted tumor had an actionable mutation based on the platforms that were used to detect mutations. And those are listed here on the graph. They had 27 sub-protocols, so treatment protocols based on these different types of mutations. And what they found when they analyzed those 27 sub-protocols is that 15% of the screened patients were able to enroll. 13% were evaluable for the goals of that study, meaning they got to a point where you could understand if the treatment was impacting their tumor, reducing their tumor size, and only 78 patients on those 27 sub-protocols had an objective response, so tumor shrinkage. When we looked at the types of mutations, P53 is right at the top, and then you can see things that you know of, the RAS mutations in adults, and others as we go down the list. And I present this to you to contrast it to what we know about the pediatric NCI-COG pediatric match study. So same platform to look at mutations in patients with relapse and refractory cancer. And Will Parsons presented this back in 2022 of the first 1,000 tumors that were screened. What's really interesting to me about this is a number of things. One is 32% of the tumors screened from patients had actionable mutations. That's very similar to what we saw in the adults with nearly 6,000 patients, and they had 38%. 28% had a treatment arm available to them, and we had 14 different treatment arms in this, and 13% of the patients were enrolled on that. We are still in the process of understanding what the response rates were to the therapies, and so that data will be forthcoming. And I know some on the phone are part of leading some of those sub-protocols, and we appreciate that effort. What's also interesting about this is, similar to what we saw in adults, TP53 was the most prevalent mutation. We also saw a lot of RAF-RAS pathway mutations in this. And so although children are much rarer than adults, the idea that some mutations in adults and drugs that are designed to interrupt that pathway can and should be used in children is very plausible, but the number of children available for those studies is small. And so because of that, the Race for Children Act was created and enacted in 2020. So RACE is the Research to Accelerate Cures and Equity for Children's Act, and it is the law that pharmaceutical companies and others involved in drug development have to evaluate molecularly targeted oncology products in children if the target is relevant to the cancer in children. So we just saw that for many of the most prevalent mutations, they across both adults and peds, but the tumor histology is very different. This therefore removes the histology-based orphan drug exception that the Pediatric Research Equity Act had put in place. And that was what was allowing drug companies to say, oh, this drug with a certain target, we're just developing that for lung cancer. And lung cancer doesn't occur in kids, so we don't have to develop it for kids. So this act obliterated that exception and loophole. The focus of this law is to accelerate the appropriate initial pediatric evaluations of early drug development, and it impacts preclinical, clinical, and clinical realms. In the preclinical space, there is a carve-out in this law that says if no preclinical data is required if substantial data exists. And that sounds great, but very rarely. First of all, how do you define substantial data and who gets to decide that? So it's a nice statement, but it really doesn't alleviate the need for very good, rigorous preclinical data in order to move drugs into children. We know that there are a growing number, and last time I counted more than 230 drugs on this list of relevant targets. And therefore, we need high throughput screening to determine which we want to move into children. And we know that pediatric cancer-specific models are needed to do that. And most of those models exist in academic laboratories and really present a very good opportunity for partnerships between pharmaceutical companies and academic laboratory and investigators to really understand what those models are and how best to test these drugs to determine if we should bring them into clinical trial. Because we know most pharmaceutical companies, not all, but most, don't have pediatric relevant models. So what's the impact of the Race for Children Act on clinical trials? As I mentioned, it is promoting histology agnostic development, meaning it's no longer about developing drugs in a single disease or a single cancer in adults. The pediatric plan is required by the FDA before a new drug application or a marketing application is filed at the FDA. And this is a really important thing because this is what drives getting drugs into children faster. The law requests an appropriate formulation or a plan to have a pediatric appropriate formulation. It doesn't mandate it. The law, when you talk to the folks who are really involved in bringing it into law for the RACE Act, it was not intended to increase the number of phase 1 trials. However, that may be an unintended consequence of the act, and it is yet to be determined if that's a good thing or not. One of the issues is that the Race for Children Act does not prioritize when there are multiple agents in a class of drugs or what to do when there are second or later classes. And I think this provides a really good opportunity for academic clinicians to really have input on how to do this. And it really implies that global collaboration is necessary. The last panel in this graph is an update that the FDA shared back in 2023, looking at what has the early impact of the Race for Children Act be? What is that impact? They see that there is a closer alignment between pediatric investigational plans, PIPs, which are the European equivalent of the pediatric study plan, which is the FDA plan for how you're going to proceed in children. They have also found that 40% of these pediatric study plans at the FDA include a plan for pediatric investigations, and there's a growing number of trials that they're looking at. The graph presented looks at the number of oncology new molecular entities or pre-drugs or agents that have planned or ongoing pediatric investigations. And as you can see from the FDA perspective, this is a very nice measure of success with significant growth between 2019 and 2021. But I do want to point out that this is planned or ongoing. So it doesn't necessarily mean that there are 80 trials currently with pediatric trials when back in 2019 there were less than 20. It means that they could be planned, but a good step forward nonetheless. So what is the impact of the Race for Children Act? It's an opportunity for earlier access to targeted agents for children. I think we can all agree on that. It does have the expectation for increased preclinical evaluations in children, and it has contemplated the need, and we have seen this both in academics, and I just showed you examples with the pediatric match study for master trials or basket trials. It implies a desire, and I think it's a real need for better industry academic partnerships and to bring in regulatory considerations early in planning for clinical trials. One of the concerns is that the Race for Children Act may be driving our industry partners to consider single trials and plan those trials rather than the development of full development plans for how you could bring this drug to meet the unmet needs of children with newly diagnosed cancer. We also see an ongoing need for agents to target pediatric cancer specific targets. And I think that is something that is going to require significant partnerships and largely heavy lifting by the academic community, not only to define those targets, but to define potential drugs for those targets, because we know there isn't a great potential for commercialization. So commercialization requires regulatory approval. And, as academic pediatric oncologist, I don't think about the financial potential gains of having a new drug approved. I'm more concerned about what is it doing for children with cancer. but that's a luxury my colleagues in industry don't have. So regulatory approval is their goal, and it's because regulatory approval indicates that the data on a drug's effects have been determined to provide benefit that outweighs the known and potential risks for the intended population. So it includes the labeling for information on dosing, formulation, and adverse effects, and it is absolutely required before you can market your drug or sell it, or in fact, move it across state lines without marketing approval. It's also important because health authorities, payers, and insurance companies closely scrutinize off-label use. And I'm guessing some of you on the phone spend some time each week trying to get prior authorization to use a drug that we consider helpful in children with cancer, but isn't actually FDA approved to be used in children with cancer. In my mind, one of the important aspects of regulatory approval that it provides the potential for more equitable, safer access to drugs. And although I've spent my career doing clinical trials, the only way to have equitable access to therapies is if you can get them out of clinical trials and get payers to help support the use of those drugs in patients without having to travel patients for clinical trials. And so let's do a little review of approval of oncology drugs. Between 1997 and 2017, the FDA approved 126 new drugs for cancer. 5% of those drugs had an initial approval that included children. This is why the Race for Children Act was brought. We know through Dr. Neal and Steve Du Bois' group that it takes 6.5 years elapsed between the first in human study to the first in pediatric study. This defines the delay in getting new drugs to children with cancer through clinical trials. In 2018, the FDA approved 36 new drugs for adults with cancer. 19% of them had individuals less than 18 years of age included in that approval, and most of those were children greater than 12. 64%, importantly, had launched a clinical trial in children by 2020. So if you got FDA approval, if a drug was FDA approved in 2018, more than half of those had a pediatric trial ongoing once that it was approved. And relevant to the Race for Cures Act, 100% of the relevant targets had a clinical trial in children when you exclude hormonal agents that aren't relevant to children. So I think that demonstrates the Race for Children Act was successful in some respects. But let's consider a few things. This timeline for approval, the 6.5 years, that's unacceptable to all of us. It's unacceptable to our patients, our patient advocates, and we really need to do better. But one of the factors, if you look carefully, is that FDA approval in adults increases the likelihood of a pediatric trial. That's just a fact, and that has to do with commercial sponsorship. We also talked earlier, and it's really important as for the pediatric community, there's first-in-class drugs, there's best-in-class drugs, and then it's our responsibility to determine what the best-in-class for children is. And this is where our preclinical work is so vitally important. Drugs that are not approved, but are of interest to pediatrics, I think we all spend a fair amount of time thinking about those. A drug that we start a clinical trial on and it doesn't have an adult indication, and they're shelved by a drug company. And there are a number of pharmaceutical partners who are now devoted to seeing which of those drugs could be out-licensed to allow us to continue development in pediatrics. So looking at what's happened since the 1950s at the FDA, this is what has driven pediatric oncologists, especially in the United States, to be very accustomed to the off-label use of drugs. So since the 1950s, the FDA has only approved a little over 50 drugs and 70 indications for cancer in children and adolescents. And that's shown in the graph here. And we can comment on the 1980s. There was a lot of stuff going on in the 1980s, but not a lot of drug development. And so what's really interesting about this graph is it's really the advent of targeted therapy in 2010 and beyond that really drove FDA approval for drugs in children. But another example of what we know about the use of off-label drugs was published by Dr. Lim, and it showed that the prevalence of off-label use of FDA-approved targeted anti-cancer therapy in patients less than 30 is 9.2%. So what this says is if you're less than 30, you have about a 10% chance that you're getting a drug that is actually not approved for you, and it may work for you, but I think it gets back to why do we need regulatory approval? 10% of people getting drugs isn't high enough, and we want them to be able to get the drug and have it approved by the FDA so we know that it is safe and effective for them. So if you were to go to the FDA and say, how do I get a drug approved? You need a randomized placebo-controlled phase 3 trial. And that's what they're going to say. However, when you look at what the drugs that have actually been approved in children with cancer, which is the first column in this very busy table, and you look at what the approval was based on, you see a pattern here. Lots of these drugs were approved on single-arm trials or pediatric pharmacology and safety data that was linked to an adequate, well-controlled phase 3 trial in adults. And I think this is really important because what in a single-arm trial makes the difference that FDA regulatory authorities would say, this is safe and effective. Part of that is the effect size. So when you look at the drugs listed here, especially the NTRK inhibitors, we knew that we saw early on in those trials that the effect size, the effect on the tumor and the patients, the benefit could easily be seen. And I think that's what has driven this ability to use single-arm trials to either get regulatory approval or support regulatory approval. And what I'd like to talk to you about in the rest of the talk is, what does it mean to take data that's not generated by a pharmaceutical company and apply it to supporting FDA approval for drugs? So globally, we know through a clinicaltrials.gov database search looking at the years 2000 to 2020 that Brahm Dewal did and published, that only 25% of interventional cancer trials recruiting pediatric patients involve an industry partner as the primary sponsor. So that is clear evidence that academic trials are the dominant type of trials in patients less than 18 years of age with cancer. However, when you step back and look in the United States at the number of children with newly diagnosed cancer who are participating in clinical trials, it has dramatically declined. And in a study that looked at it in the time frame from 2024 to 2015, it was as low as 20% of those newly diagnosed patients. That compares to over 70%, 2 decades before that. So the question is, there's a lot of reasons contributing to that, but I think our commitment to clinical trials and moving drugs rapidly and effectively into the upfront setting is really important. So at this point, I would like to see if we could have you raise your hand if you would like to share any experiences comparing the differences between clinical trials you may have been involved in or conducted that were industry sponsored, cooperative group sponsored, or investigator initiated trials that you have led. So anyone want to raise their hand and sort of, what are the main differences that pop to mind when you think about those 3 different ways to run a study? What does it mean for your site? And while we're thinking, and please pop in whenever you can, but I will say that what we're going to talk about in the rest of this talk is really why there are differences. And, I can say, if you raise your hand, we'll unmute you and you can share your experience. We're happy to have other people talk other than me. And please jump in as soon as you're ready, but I'm going to go on to the next slide to sort of frame this question a little bit. So when we think about investigational new drug applications, INDs, that's the agreement that you come to with the FDA to allow you to put a drug into any person, including children. There are 2 big types of these things. One is commercial INDs, The other is research INDs. So commercial INDs are held by the sponsor, who is a corporate entity, and the primary intention is to commercialize. I see Joanna has raised her hand. Let's see if I can unmute Joanna.
I unmuted her, Beth. She can
Thanks
talk.
so much. Joanna, jump right in here.
Hi, thanks, Beth. I was just thinking about how to frame it. When I've worked with industry partners, as you're showing here, their focus is, okay, how are we going to get the FDA to approve this drug? You're talking about a very small patient population, and what happens if something terrible happens in children that will affect our FDA approval. And so there's a lot of hesitation, and there's a lot of decisions of their of my concept moving forwards with them based on what their adult development strategy and their data is looking at. And then I also think that just as a site PI here at a phase 1 center, looking at industry-sponsored written trials versus like COG written trials or other academic institutions, the things that they ask for is sometimes in terms of studies required or reporting is sometimes very different between the two groups, which probably goes to your point here in terms of like, what is their end goal?
Exactly. And we'll go through that and hopefully get a little bit of perspective on why. And my goal is really to help us understand, is there a middle ground that we can get to? Because as I said, I'm not saying we shouldn't have regulatory approval for drugs. What I'm saying is, when we have a commercial sponsor whose intention is to commercialize, that's great. That's what they do. That's why they exist. As Joanna mentioned, what they expect out of that trial is to meet and probably perhaps exceed anything that the regulatory authorities, the FDA or EMA, might ask for. So they're in a very proactive mode. And what that means is you end up with very complicated data collection. And that's why the monitors come to your site every 6 weeks or every soon or more often than that, and there's all sorts of queries to clean the data prospectively. All of that is driven by this idea that they want to commercialize. They want to put this data and have it be of regulatory quality. In academics, what we're doing, the sponsors of research INDs are individual investigators, academic institutions, or nonprofit entities. And we go into these INDs with no intention to selling the drug, to commercializing the drug. And we're doing this for research results, publication, and the betterment of the care of children with cancer in our case. This is accomplished by submitting a letter of authorization or cross-file to the drug company on their commercial IND and saying they'll let us use that, refer to that to do our IND. This is very different, and this is one of the main challenges to, can we as academic institutions develop drugs in our laboratories and take them all the way through the commercialization process? And we'll touch on that as we go through. So there's a fundamental difference in the aspect of allowing us through the FDA to put agents into children or adults for that matter. I want to point out this example of larotrectinib, a very highly successful NTRK inhibitor, and talk a little bit about how this came to be. The first study was conducted with a commercial IND by Loxo Bayer and treated the full pediatric age spectrum as a dose finding and objective response rate study. And they ultimately got a highly successful age and histology agnostic FDA and EMA authorization. So that's a commercial IND doing what a commercial IND does best. The NCI also had a research IND that's ongoing in part of the pediatric match, where they're treating patients with relapse and refractory cancers and looking at the objective response rate. Importantly, Ted Leitch has run a study in the Children's Oncology group where there was a research IND held by the COG and a COG group-wide study to look at patients with newly diagnosed tumors that harbored NTRK fusions. And this is the study that the drug's FDA approved. You can get the drug, but we actually don't know how to use the drug. You can give it, but what do you do? When you start it, when do you do the surgery? What happens after you do the surgery? Do you restart it? How long do you restart it? If you stop it, do they recur? This study conducted by the COG is going to ask those questions. So I see this as a really nice example of, no matter what type of IND you're doing, keeping your understanding of how to meet the unmet need is critically important. So when we look at how the COG has done in supporting FDA labeling, they have a large number of drugs in which COG data has been used to support FDA filing. So research INDs that ultimately the data was pulled out of and used to support filing. I'm going to go through a very successful example, dinutuximab. The NCI cooperative group COG did a phase 3 randomized trial of this immunotherapy with, humanized anti-GD2, not humanized, chimeric anti-GD2 antibody in high-risk neuroblastoma patients. It was sponsored by the NCI, a research IND, initially manufactured by the NCI, and then by a company. And the study was activated in 2001 and treated 226 patients. And I'm sure you're all aware of the landmark paper by ALSF that demonstrated that this immunotherapy with anti-GD2 directed therapy was superior to the standard therapy with isotretinoin, both in event-free and overall survival. So that's a great academic success. It's a success for our patients. It was also a regulatory and industry success because it was that data that was used to, in 14 years later, get FDA approval for dinutuximab. And regulatory issue is that United Therapeutics received a priority review voucher for this pediatric thing. So it's one of the incentives for doing pediatric drug trials. But what did that take? What happened in those 14 years? Those 14 years included 19 amendments to that primary phase 3 trial. We knew that the adverse events were collected in a way that was very good for making sure patients were safe and standard for COG and NCI trials, but we didn't collect all of the toxicity data. In addition, the toxicity grading criteria changed from the common toxicity criteria version 2 to an entirely new, different, and upgraded, more granular toxicity reporting, toxicity terminology criteria for adverse events, version 4. So we had to do a lot of mapping to understand how patients treated early in the study toxicity related to those treating later. And as I mentioned, the manufacturing of the product changed from the NCI to a pharmaceutical group. That pharmaceutical group made it clear they were going to commercialize this and use this data. They elected to do an additional trial that was a comprehensive safety trial that in a 3-year period enrolled another 105 patients. And they did exactly what Dr. Yee mentioned. They collected a ton more data. The sites collected data in a way that was much more granular and more aligned with regulatory approval. What happened to get this drug FDA approved, some of you on the line may have been part of. It's known as the Great Retrofitting of Data experiment. And it is something that none of us really want to relive. But we did an extensive retrospective data mapping and attempts to harmonize the data, and were successful enough to get it FDA approved. We also had to send monitors out to sites to look at that data retrospectively and prospectively, and ultimately the FDA inspected both the operations and site activities. And so this was successful, but the effort that this took was something that both academics and industry partners said, wow, that was a lot of effort. Glad we were successful, but what can we do moving forward? So another model is the model of entrectinib. And this is where the importance of Alex's Lemonade Stand and the Centers of Excellence comes in. So entrectinib is the second-in-class oral NTRK ROS1 and ALK inhibitor. And through the Alex's Lemonade Stand Centers of Excellence academic collaboration using the infrastructure grant and the expertise of our scholars, the Star Trek Next Generation phase 1/2 trial of this drug was launched in 2016 by a very small biotech company that held the commercial IND. But the trial was designed and led by Ami Desai, an ALSF scholar, and it initially was only 4 sites. CHOP, Dana-Farber, Baylor, and UCSF, and got off the ground pretty quickly. The drug was acquired by a large pharmaceutical company, Roche, and transitioned this trial, because of its success and what we learned, into an international 40-site, 11-country trial. And what we learned along the way, that there was an adult recommended dose, and this was very successful because I'll show you, ultimately ended up, as you know, in FDA approval. But how did we get there? So we knew that entrectinib was going to be active. We knew that it was a second-in-class molecule. We elected to do a dose, a 3+3 maximum tolerated dose toxicity study Because it was 2016, we would have had to get an IND to determine, NTRK fusion status on the patients, which we weren't ready to do. And knowing it was second-in-class agent, we knew that if we biomarker selected, we would have much fewer patients. And at that time, scientifically and ethically, it was still ethical not to biomarker select. So we didn't. And we ran this dose escalation trial in ultimately 15 patients, and determined a dose that we could move forward and turn the trial into a biomarker selected trial at that point. What we found was that the maximum tolerated dose was much higher than what we would have predicted based on the 600 milligram adult dose, which would have been somewhere between this first two dose levels. But these dose levels were actually highly constrained by the fact that there were only two capsule sizes available, 100 milligrams and 200 milligrams. The size of these capsules was size 0, which means the capsules themselves were about 2 centimeters. And when you think about treating an infant with infantile fibrosarcoma, that is not going to work. So there were also some notable non-dose limiting toxicities that we uncovered using this design. What Roche did was they took the detailed pharmacokinetics and they modeled it again with their adult data. And they found that by PK modeling, 300 milligrams per meter squared was actually equivalent to the exposure. That would have been very similar to what we would have loved to do of 350 milligrams per meter squared, but couldn't because of their formulation constraints. And they did go on to develop an oral liquid formulation. But this is how you work together to get drugs FDA approved. This is the swimmer plot that looks at the different fusions of interest and how importantly they had prolonged disease stabilization, and in some cases partial and complete responses. These are the scans in various patient populations that were really dramatic objective responses, not only in children with brain tumors in the first panel, but also children with melanoma, infantile fibrosarcoma, and inflammatory myofibroblastic tumor. So as I mentioned, there was FDA approval 3 years after the pediatric study started in 2019 for children greater than the age of 12, based on using the safety and PK data from that initial trial that Alex's was so instrumental in getting off the ground. We were able to get to that. EMA authorized this in 2021, and the FDA, the company came together and got a pediatric formulation, and the FDA has now approved this drug down to children greater than 1 month of age. So what is the impact of formulation? And I'm going to go through this because I want to make sure we have enough time to have some questions. Larotrectinib had an oral liquid formulation from the very beginning. They did a biomarker selected study, and the median age was 4.5 years for the 24 patients who were enrolled. In Ami's study, we had these very big capsules, and therefore our median age was actually 10 years of age. And we did an MTD study. So this really demonstrates to me that the impact of being able to get a drug FDA approved in children less than the age of 12 is critically dependent upon the formulations you have available. And so one other model for how to partner is something we're doing at St. Jude currently in the ONIT study, which is a study of the liposomal irinotecan product Onivide with either talazoparib or temozolomide, and Sarah Federico runs this study. We are holding a research IND to run this study, and we knew from the beginning we had 2 pharmaceutical partners on this study, and they had very different goals. One stated the intent to use the data for regulatory purposes. So how can you prospectively plan for a research IND data to be used for regulatory purposes? And you do that by shoring up your infrastructure and negotiating with the company for what are the guidelines for the type of data that they are going to want to see to put in front of the FDA, and what do you have to do in order to do that. So there were lots of things that we did, including centralized response assessment, increasing monitoring at all sites, making sure we had Part 11 compliant databases that align with FDA regulatory expectations. And we pre-negotiated database transfers and resource allocation to support all of these things under a research IND. This study is ongoing, but I can tell you that the partnership has been phenomenal in making sure we're meeting the expectations for the data in a way that is responsible, and that we have resources allocated appropriately to do that. So these are trial perspectives, and I think we've touched on all of these. This is a slide that Ellie Barry shares with me. When we think about academic trials, we often have limited experience with intent to file. We have reduced source data verification, meaning we don't monitor our data as carefully as industry might. And we collect what's required to ensure patient safety and good publications and demonstrate benefit. We don't collect more than that because it's very expensive. We're often pragmatic and focus on unexpected and serious adverse events. And we're really looking to benefit patients and publication, not commercialization. Our industry partners have the intent to file. This means they do rigorous data management collection plans with full monitoring. They collect anything and everything. I mean, those of you who've participated, they ask you about every chemotherapy and the dates that the patient ever received, and they objectively verify all aspects of trial conduct. They anticipate the filing requirements and maybe over-anticipate them because they don't want to have to go back and they want to be able to answer the FDA. And they file their application as how they communicate their results. So I'm not going to touch on this slide too much, but except to introduce you to the International Council for Harmonization of Technical Requirements for Pharmaceutical Human Drug Use. And this is the guidelines that both the EMA and the FDA use. And this touches on all aspects of getting a drug into patients and really outlines very specifically what is required to do that. And it's these non-clinical study reports here in the green module 4 that is where all of this safety and toxicity data in animals gets put. And those are things that often fall in the gap between what our discovery scientists and our academic labs want to do and those of us who run clinical trials want to do. So the Accelerate Fit for File working group was set up in 2019, and it really, the goal was to bridge the gap. What could we do? Could we have conversations and partnerships to understand how can we meet the needs of regulatory requirements with academic trials and come part way? So this included academicians, industry partners, regulatory folks, and patient advocates, and ultimately ended up in a 2022 publication about how we got to some recommendations. Importantly, there is also Accelerate's educational platform, Aladdin, has a 3-part webinar that we put together that for those who are interested is worth thinking about how to do this in clinical trials that are run by academia. It's 3 parts. The first is, should and could my trial be fit for regulatory filing? The second is, a spoonful of data helps medicine go down, purposeful collection of clean and reproducible data in a trial. And with great need comes great responsibility, delivering high quality data and successful filing applications. So I think this is a critical thing for us to consider. What really are the unmet needs in childhood cancer? And this is a very important publication by Gil Vassal and Teresa de Rojas. And what's shown on this graph is on the x-axis is the proportion of deaths as a % of children in the European Union who died of cancer in and around 2007. And what's on the y-axis are the common and other pediatric cancers. And they're divided up by the number of approved drugs since 2007 by these diseases. So for example, lymphoid leukemia, we know, accounts for almost 25% of the children who die from cancer. And there were 2 drugs approved for leukemia in this time period. But as you can see, there are also 2 drugs approved for thyroid carcinoma, and you just saw 2 drugs approved for NTRK positive malignancies. And so those 2 cancers together don't account for much of the proportion of death. So what this analysis showed, where there were 16 drugs approved for children with cancer, 8 of them are indicated for cancers that altogether account for 5% of childhood cancer-related deaths. And I think when we talk about unmet need, this is the gap between regulatory approval for things you can target and get approval for, and what we as clinicians know to be the gap, treating children with difficult to treat cancers. The other gap that we have is we know that cancer happens worldwide to children, and there's a great divide depending on where you live. But more than 400,000 children are diagnosed with cancer each year, and more than 110 of those children will die due to that cancer each year. We know that the 5-year overall survival, as we saw at the beginning of the talk, for children in high-income countries is greater than 80%. However, in low and middle income countries, the number of children who survive for 5 years with a diagnosis of cancer is less than 30%. So this opens up the opportunity for really considering unmet needs. One of the next steps for entrectinib, we hope to bring entrectinib to low and middle income countries through a St. Jude sponsored trial in a partnership with Roche. And we really see this as a major step forward for considering unmet need throughout the world. So finally, childhood cancer is rare. Clinical trials with biomarker selection further limit the population of eligible patients. The role of academic clinicians is critical. We have to consider the types of therapeutic interventions, including combination therapies, multimodality therapy. It's not just about small molecules either. We've got CAR T-cell therapy, radiation, and surgery to consider. We also need to be very aware of the evidence for drug target and response relationships, the toxicity profile and the reversibility of those toxicities, and the developmental considerations, because our patients are growing and developing as we're treating them for cancer, and as we've shown, formulation. So what should we do? Regulatory approval is important for safety and equity. Early, robust, multi-directional partnerships among academic investigators, cooperative groups, regulatory agencies, and industry are necessary to move the needle for childhood cancer. The intention for regulatory filing should not and may not be known during trial development, but that doesn't mean that data from clinical trials can't be used. Those data from clinical trials conducted under research INDs can be used to support regulatory filing. I would like to say that we would prefer not to try to retrofit data like we did in the dinutuximab example, but perhaps think more prospectively, partner, and do what we can to improve the quality of data in the eyes of regulatory authorities without completely making it impossible to conduct a research IND study because it's too expensive. The role of the academic clinician scientist is to ensure that trials address the unmet need of children with cancer, and if those trials show promise and are successful, lead to outcomes that change the standard of care. So with that, I want to thank you for your attention. I want to thank Alex's Lemonade Stand Foundation and the Centers of Excellence for the support of infrastructure, because what I just talked to you about was the infrastructure that supports the science behind things. The Accelerate Group, COG, CTEP, our industry sponsors, regulatory agencies, patient and family advocacy is critically important to moving the needle. And I have a sincere gratitude to the patients and families who participate in clinical trials. So thank you very much.
Thank you, Beth. We do have one question for you from Liz. I have heard concerns from physician scientists that a company will take on a therapy that's effective in kids and decide to stop producing it if it does not prove to be effective for adults. Is this a real concern in your experience? And if so, what can academia do upfront to avoid that happening?
Yeah, Liz, it's a great question, and it is a reality. And it is based on this fundamental divide in not the people who we talk to about developing clinical trials who are in industry, you know, our scientific partners, our medical folks in those companies who think about pediatrics. It becomes a business decision at some level when you're in a for-profit company. And we're not going to get away from that. And I think the ways to mitigate that are tricky because they are fundamentally there to get regulatory approval. And as you heard Joanna and others have experienced, you know, we don't have a patient population that easily gets them regulatory approval. And so what do we do when a drug gets quote unquote shelved? And it happens frequently. It happened here at St. Jude in the last month. We had a drug that we had a trial ready to go for, and suddenly the drug went away. There are companies that are beginning to form more than one. I mean, a couple come to mind quite readily that are really focused on pediatric drug development. And what they're doing is out-licensing those drugs, meaning they develop a contractual relationship with the main pharmaceutical company to say, we're going to develop this in pediatrics. Those are very complicated relationships, because they're still companies and they're still based on the economics and getting drugs approved. But companies like Day One Pharmaceuticals, SpringWorks, and others are in the business of doing that. And I think those are great partners to have because they do think about it. I think the other challenge, Liz, is that most pharmaceutical companies are not interested in pediatric specific targets, things that we know we need for DIPG, Ewing sarcoma, other unmet needs, because there's no market share for that. So developing and working together to be able to take drugs from bench to bedside and then have perhaps spin those off to companies when we have successful endeavors so that it can be commercialized, or figuring out ways to commercialize those through academic pathways and partnerships, I think is the way forward. It's not simple. What I've laid out today is a step in the right direction. Can we, as people who conduct clinical trials, begin to get our data in shape so that the regulatory authorities are like, yeah, this is good data. This is supported and backed up in a way that we know it's good enough to support their primary mission, which is the safety and benefit ratio that can be objectively seen by regulatory authorities.
Thank you. If anyone has questions for Beth, you can put them in the Q&A, or if you would like to ask, you can raise your hand and I can unmute you. Okay. I have one question as an extension to your response to Liz's question, and that is, within academia, if there is a target that the pharma is not willing to develop a drug for, is there anything that can be done to bridge that gap between finding the target and actually developing the chemistry to find a drug for that target?
Yeah, that is a great question, and I think it speaks to the divide between discovery science and can we get to a lead compound. I think we can, and I think that's been shown over and over. Then we have, on the other end of the spectrum, many of us who focus on, can we get clinical trials done? What does it take? And we have that expertise. What we don't have is the middle piece, which is often referred to as investigational new drug application enabling studies. And those are all those things in that green box I referred to in the good clinical, the good laboratory practice and the way to get trials through IHC and for regulatory purpose. Those IND enabling studies are things like animal toxicology in 3 species. How much of that do you need to do? Pharmacology, drug-drug interactions, all sorts of things about stability and formulation and making sure that the compound you're going to put into patients does what you think it's going to do in a preclinical setting. All of that, which I bundle is called in IND enabling work, it's not the type of science that discovery scientists get rewarded for. It's also very expensive, but it's necessary. And so can we get to a place where we can figure out how to work together to, first of all, maybe reduce the amount of that needs to be done, or figure out novel mechanisms to be able to resource that appropriately for highly selected, highly promising agents. I think it's something that I'm committed to, and a number of places are really looking at what does it take to truly take something from bench to bedside and to commercialization in an academic setting. And it is clearly expensive. And, those of us who have heard what it costs a drug company to bring a drug to market, it's astonishing. And part of me asks why. And, it's hard not to think this is because it's being run by a billion, the global pharmaceutical industry is a $1.6 trillion industry. Only 7% of that is dedicated to pediatrics. And when I say pharmaceutical industry, that's everything from, over the counters through oncology drugs. But that industry is supported by billion-dollar industries, not only in pharmaceutical companies, but contract research organizations, regulatory approval organizations, and the people who are consultants to those. And one has to ask the critical question is, are those studies generating too much data? I think that's what we're trying to get to with what does it mean fit for regulatory purpose and how much data is necessary.
Thank you. Amit has a question. I'm going to unmute him. Amit.
Great, thanks so much. Beth, great talk. I just had one question to sort of touch on a point you made earlier in the conversation about the Race for Children Act, sort of creating a little bit of a conflict with figuring out kind of what's the best in class for kids. And I think you had suggested that sort of fall to preclinical studies. And I think where, you know, there's different potencies of drugs, that's clearly something that can be figured out. But otherwise, this seems like a really tricky issue to try and resolve. And I was just wondering if you could comment a little bit on what sorts of studies are needed to really get to that answer.
Yeah, I think that's it's a great question. My thoughts on it are we almost never put all our eggs in one basket in the pediatric space, right? If there are multiple drugs in class, we rarely say no until it gets to the point of how many studies can we actually open. And that's because oftentimes we're putting these drugs into clinical trials before we have all the information. And, you know, there could be later side effects that turn up that make it less agreeable to treatment of children. We may learn of toxicities in children that are not expected and we can't find in adults. And those sorts of things impact what I consider best in class in children. Predicting what those are is really hard. You know, drug-drug interaction studies help in some respect, but in some ways you have to be willing to project. I think one of the major things that I think about and tried to touch on is this idea of formulation, because part of this, and you know, what drives toxicity? It's how much drug is in the body. And so the idea that we need to provide dosing that is appropriate for children and be able to deliver it in a way that they'll actually take the drug and not spit it out is an important thing, but also so that we're not treating above the targeted dose that we need, because then all we're getting is side effect in addition to the benefit. And that's where we really want to concentrate. So best in class is a tough thing to get to, but it's not usually best in class for children may not be first in class, but that doesn't mean we're not going to go after the first in class to get into children. Not a great answer to your question, but a little bit of a philosophy on how we get there. But that's what I'm looking for, is not necessarily what's first, but what's best and why, and can we begin to decipher that?
Thank you. I think that was all the questions we have. Thank you, Beth, for sharing with us today. Really appreciate it. And thank you, everyone, for attending.
Thank you. And thanks all for dialing in. You can feel free to email me if you have questions. My emails are out and about. You can easily contact me. And again, I really want to thank Jay, Liz, everyone at Alex's Lemonade Stand for not only the support for pediatric oncology, but also the ability to present this talk today. Thank you.